Okay, so in summary, you'll have to extract, like, first of all, a square window, which is called a block. Okay, then you will divide that block into smaller cells. They could be 2x2 or any other shape as well. Then you compute orientation histogram for each cell. You concatenate like 4 histograms. If you have 2x2, you will have 4 different histograms. And then you will perform normalization, and that's it. That's your HOG histogram algorithm. Okay, so now there are like different set of parameters which actually play a very crucial role how good the features will be. So one of the parameters is how many bins should you use. Okay, so in the example we discussed, we were using 9 different bins, but of course you can use like more bins or fewer bins as well. So you know that your range is from 0 to 180 or it's 0 to 360. So you can create like bins of, let's say, 10 degrees, or it could be 30 degrees or 40 degrees, right? So that's one parameter that will control what should be the size of the histogram, right? And that will control like what will be the size of the feature at the end. So if you're using 9 bins, which is usually the case, then each bin will cover 20 degrees. Cell size, like on the actual image, usually it's used 8x8 pixels. Okay. And block size is usually used 2x2 cells, which means like each block will have 16x16 pixels. Okay, so if you use these configurations, and of course these are like some parameters which can change, but if you use this, then your HOG feature vector will be 36-dimensional long, because for each cell you will have 9 different values coming from the histogram, and you have 4 different cells. So this turns out to be 36. And depending upon like how many blocks you have in your image, just multiply with that number. That will give you the full length of your feature vector. And this is like one visualization of how HOG features actually look like. You can see that it's kind of giving you a good visual, right? What is present, you can say that, okay, this seems like a woman standing here. So this visualization, I think the way it was done is, for each cell, you have the histogram, right? And then you pick like the orientation which is like more dominant in that cell, and that orientation is actually drawn for that cell, and that's how you get this visualization. Okay, so not exactly HOG histogram, but a way to visualize that. So this will actually help you in debugging like whether your extracted features are meaningful or not.